Imagine a bustling highway where information zips along at lightning speed. This is the journey of a nerve impulse as it crosses a synapse. When an electrical signal reaches the end of a neuron, it triggers the release of neurotransmitters, tiny chemical messengers. These neurotransmitters float across the synaptic cleft, a microscopic gap, like cars merging onto a new road. Once they reach the receptor sites on the next neuron, they bind like a key fitting into a lock, opening channels that allow ions to flow in. This influx generates a new electrical signal, continuing the message down the line. It's a precise dance of chemistry and electricity, ensuring our bodies respond to stimuli in real time. This intricate process not only allows us to move, but also shapes our thoughts and emotions, making every moment of our lives a symphony of neural communication. So today, our topic is about transmission of nerve impulses across and at the axon terminals of free synaptic neurons. The cytoplasm of kinetic nerves contains numerous spherical sacs called synaptic vesicles. Each of these vesicles has as many as 10,000 molecules of neurotransmitter substances. The arrival of an action potential at the presynaptic plasma membrane opens voltage-gated channels that allow calcium ions to flow into the synaptic terminal, releasing the neurotransmitters. The neurotransmitters then diffuse across the synaptic cleft and bind to the receptors on the postsynaptic membrane. Binding of neurotransmitters to the postsynaptic neuron receptors opens channels and allows sodium ions to diffuse across the postsynaptic membrane. As a result, it is called excitatory postsynaptic potential, EPSP. At other synapses, different neurotransmitters bind to channels that are selectively permeable for only potassium ions or calcium ions. When these channels open, the postsynaptic membrane hyperpolarizes. Hyperpolarization produced in this manner is called inhibitory postsynaptic potential, IPSP. Various mechanisms rapidly clear neurotransmitters from the synaptic cleft, terminating their effect on postsynaptic cells. Certain neurotransmitters may be actively transported back into the presynaptic neuron to be repackaged into synaptic vesicles, or they may be transported into the neuroglia to be metabolized as fuel. Other neurotransmitters are removed from the synaptic cleft by enzymes that catalyze the hydrolysis of the neurotransmitters, like acetylcholine, which is hydrolyzed by acetylcholinesterase, and adrenaline, which is broken down by monoamine oxidase.